Hello everyone, my name is Christian Stevens, and I am currently a PhD student in the Plant and Soil Science Department at Texas Tech University.
And my name is Maximus Gudino. I'm an undergraduate student researcher, also in the Department of Plant and Soil Science here at Texas Tech University.
And today, we will be walking you through the step-by-step procedures in the artificial cross-pollination of rice.
This video is going to be cut into 3 separate sections. The first is going to cover the materials we'll need. The second section is going to cover the emasculation process. And the final section is going to cover the cross-pollination of rice.
As you may know, rice is a self-pollinating species with the anthers and the stigma inside the lemma and polea. Today, we will be crossing this self-pollinating species. We will be using 2 separate germplasm for this experiment. The female parent will be Presidio, a Texas inbred rice cultivar. The male parent will be an F2 progeny derived from a cross between Presidio and ST12, which harbors 2 very important yield determinant genes, WFP and GN1A. When performing artificial cross-pollination in a self-pollinating species like rice, it is necessary to determine which parent will be your female recipient parent and male donor parent. In this case, we will be crossing the cultivar Presidio with an F2 progeny harboring genes that increase yield potential. The goal will be to produce progeny that have the background of Presidio, but the genes from the F2. Here is an example of a portion of the panicle that is already self-pollinated. As you can see, the anthers have emerged from the spikelet, which is a sign that it has already gone through anthesis and has flowered. We will not use this upper 1/3 portion of the panicle for emasculation, so we will trim these portions off of the panicle. To begin, you must open the leaf sheath to expose the entire panicle. Once opened, you can see the immature portions of the panicle. This bottom 1/3 portion is not yet ready for emasculation and can be removed. An initial indicator of this immaturity is in the anther position. If the anther is below the 1/3 section of the spikelet, it is not ready for emasculation. Another indicator would be its misshapedness and pale color in comparison to the mature panicles. I will here demonstrate how to remove the branches off the panicle that contain immature spikelets. Our aim is to select the middle 1/3 section of the panicle, cutting off all spikelets that are either pollinated or immature. In order to extract the anthers from the spikelet, we must be aware of the anther position when making cuts into each individual spikelet. The aim is to cut the spikelet diagonally above the anther position to efficiently remove them without extracting or damaging the stigma.
Rice spikelets will have a total of 6 anthers. When removing the anthers, aim to remove all 6 from the side of the spikelet for an easy extraction. Do so very carefully. Again, be careful not to damage the stigma. Remove 3/4 of the flag leaf from the panicle. This will allow you to place the flag leaf inside of the bag alongside the panicle. Photosynthesis in this leaf provides most of the carbohydrates needed for grain filling. Select a male donor parent that has reached anthesis. When the anthers are a distinct yellow color, the pollen grains are ready to be dusted onto the female recipient parent. Under warm, humid conditions, rice has pollen. The prime time for dusting would be between 11 a.m. and 12 p.m. in the greenhouse when the temperatures have reached their peak. Take the panicle of the male donor parent and gently tap it against the emasculated flowers of the female recipient parent with enough force to dust the pollen grains onto the female panicle. Cover the entire panicle, including the flag leaf, with your glassine bag to prevent exposure to other sources of pollen. Make sure to write clear, detailed information about the germplasm utilized in your cross. After around 7 days, you can check your panicles for early seed set. This panicle is at its peak for harvest maturity, which is at 3 to 4 weeks after pollination. Cross-pollination was indeed successful.
Thank you for joining us. We hope you have found this journey through cross-pollination of rice informative and engaging.
Stay tuned for our next video, where we will explore marker-assisted selection in rice breeding. Happy breeding!